Hey everyone, and welcome to This Explainer. Today, we're looking at an absolute masterclass in biological engineering. We are going to unpack ELISA, the enzyme-linked immunosorbent assay. But look, I really don't want you to just think of it as some standard run-of-the-mill laboratory test. Think of it more like an incredibly elegant, microscopic scale, one that actually lets us reach into a chaotic biological soup, grab a single, highly specific protein, and measure it with pure mathematical precision.
Okay, let's just dive right into this. The blueprint you're looking at here shows you the core mechanism of the entire assay. It is literally the perfect marriage of two distinct biological powers. Over on the left, in navy blue, you've got this specificity lock, an antibody that's engineered to grab onto one and only one specific target. And then on the right, in gold, you have the enzyme signal amplification. It's an attached enzyme that acts kind of like a molecular megaphone, broadcasting the fact that our target has been successfully captured.
So here's a quick look at our roadmap. Section 1 covers the core concept and antibody structure. Section 2 explores the 4 main detection modes. Then in Section 3, we'll get into lab workflow and color chemistry. Section 4 tackles data interpretation and the standard curve. And we'll wrap it all up with Section 5, breaking limits and perfect synthesis.
All right, Section 1, core concept and antibody structure. To understand the whole machine, we really have to understand its primary building block.
Picture this. You have a highly complex biological source. Maybe it's serum, blood plasma, or cell culture media. That stuff is absolutely packed with thousands of different random molecules. Well, ELISA acts as a super specialized filter. Whether you're hunting for specific cytokines, hormones, or antibiotics, this assay uses immune targeting to pull exactly what you want right out of that messy mixture. And once it isolates it, it uses that enzymatic signal amplification to essentially weigh it on our molecular scale. Now, the success of this whole filtration process, it rests entirely on 2 massive pillars: specificity and sensitivity. Specificity is just the assay's ability to completely ignore the thousands of background molecules and bind exclusively to the target. Sensitivity, on the other hand, is its ability to actually detect that target even when it's present in vanishingly tiny amounts.
Now, what's really fascinating about this slide is the actual physical structure of an immunoglobulin G, or IgG antibody, because that shape dictates those 2 pillars. Notice this classic Y shape right here. The top arms, what we call the Fab region, are where the targeting potency lives. That dictates exactly what the antibody is going to capture. But look down at the stem, the Fc constant region. This lower part has highly conserved species-specific sequences. And as we'll see in just a minute, this Fc region acts as a totally crucial universal binding target for secondary antibodies.
When you harness that unique antibody structure, you unlock the 4 pillars of ELISA's utility. First, extreme specificity, which is usually driven by monoclonal antibodies binding to singular targets. Second, high sensitivity, driven by enzymes amplifying a single microscopic binding event into a highly visible signal. Third, exact mathematical quantification using optical density. And fourth, standardized scalability, universally adapted to 96 or 384 well microplates for high-throughput lab processing. Moving right along to Section 2, 4 main detection modes. Let's see how scientists arrange these antibodies. Here is the architecture matrix. Depending on what you actually need to do in the clinic or the lab, ELISA is generally set up in one of 4 specific ways. The direct method is really fast and often used for hybridoma screening. Then you have the indirect method, heavily used in serology, think HIV tests. The sandwich method is essentially the gold standard for clinical biomarkers. And finally, the competitive method, which is incredibly useful for testing pharmacokinetics and affinity. Let's actually zoom in on that indirect method for a second. See, creating a custom enzyme-linked primary antibody for every single test, that is incredibly expensive and extremely time-consuming. So labs do something really clever. They use an unconjugated, highly specific primary antibody to go in and find the target. Then, they introduce a mass-produced, way cheaper secondary antibody, like an anti-mouse antibody, for example, that specifically targets that conserved Fc region of the primary antibody we talked about a few moments ago. And this brilliantly illustrates the sandwich method, which is absolutely vital for clinical diagnosis. In this setup, you use 2 antibodies, a capture antibody on the bottom and a primary antibody on top, literally sandwiching the target protein. But here's the critical rule. they have to target completely different structural regions, known as epitopes A and B, on the target antigen. If they targeted the exact same spot, they'd just compete for space and the whole assay fails. So by requiring two distinct binding events, you drastically increase the baseline specificity of the test.
All right, on to section 3, lab workflow and color chemistry. Let's shift from molecular architecture to physical bench chemistry.
Translating this to the real world, this whole microscopic process takes hours to physically complete at the bench. It starts with coating the plate with an antigen or capture antibody at 37 degrees Celsius for an hour or two. Then comes a blocking stage, followed by really carefully timed room temperature incubations for the primary and then secondary antibodies. And between every single one of those steps, there is a crucial washing phase to remove any unattached elements. Anyone who's worked in a biology lab knows this repetitive washing basically takes up your entire day.
Just to really lock that workflow into your brain, step 1, coat the plate. Step 2, block the empty spaces. Step 3, add the primary antibody to find the target. Step 4, add the secondary antibody to bring in the enzyme. And step 5, finally, add the substrate.
Now that final substrate step, that is literally where the magic happens. Commonly, a horseradish peroxidase, or HRP, enzyme is attached to the antibody. When you add TNB substrate and hydrogen peroxide, the enzyme triggers this continuous catalytic reaction that generates a vivid blue color. And the longer you let it run, the stronger that blue signal gets. Once you're ready, you instantly terminate the reaction by dropping in sulfuric acid. That chemically shifts the color, straight from blue to a bright, easily readable yellow.
Which brings us right to section 4, data interpretation and standard curve. Time for some standard curve math.
So here's the big question. It's one thing to say, hey, this well is a darker yellow than that well. But how do scientists actually translate the specific shade of that yellow liquid into an exact, quantifiable concentration of a protein?
Well, the crucial point here is the standard curve. Scientists will run known concentrations of recombinant proteins, like, say, synthetic human IL-6, at exact serial dilutions. By reading the optical density of these knowns, they plot a linear regression line, aiming for an R-squared reliability of over 99%. Once you've got that beautiful mathematical line, you just take the optical density of your unknown sample, map it against the curve, and boom, you have your exact concentration. But remember, because this readable window is pretty narrow, biological samples often require really precise serial dilutions, sometimes 10x or even 1000x, just to make sure they fall onto this scale.
Finally, let's jump into section 5, breaking limits and perfect synthesis, because sometimes standard just isn't enough. Think about this for a second. What happens when the biological target is incredibly scarce? Sometimes the protein you are looking for in a patient's sample is so highly diluted that the standard HRP and TNB reaction just isn't sensitive enough to generate a readable yellow signal at all. You essentially need a way to turn the volume way up on that molecular megaphone. The solution they came up with is just incredibly elegant. It's called the Avidin-Biotin complex, or the ABC system. Instead of binding just one single enzyme to the secondary antibody, scientists attach biotin. They then introduce streptavidin, which is this molecule that has exactly 4 highly receptive slots for biotin. One slot binds to the antibody, which leaves 3 slots completely wide open to capture additional biotin enzyme conjugates. Just like that, you have effectively tripled your enzymatic signal for every single target found. It's brilliant.
Let's move to this comparison and see how this builds into the overall performance limits of detection. The indirect method is super reliable down to nanograms per microliter. The sandwich method, again the clinical gold standard, pushes that sensitivity way down into picograms. And if you swap out that color-changing TNB chemistry for light-emitting chemiluminescence read by a luminometer, you can actually detect absolute trace amounts down in the femtogram range.
Which really brings us to the ultimate synthesis of this explainer. The enduring dominance of ELISA relies entirely on this one perfect pairing. On one side, you have the elegant, atomic-level targeting mechanism evolved by the mammalian immune system, the specific key in the lock. And on the other side, you pair it with the raw, mathematical power of enzymatic chemical amplification, the megaphone.
It truly is a masterclass in biological engineering. By simply observing how our own bodies naturally identify microscopic invaders, scientists were able to isolate that mechanism, strap a chemical amplifier to it, and completely transform modern medical diagnostics and research. Honestly, it makes you wonder, What other profound technologies are hiding right now in the basic biology of our own immune systems, just waiting to be discovered and scaled? Thank you so much for joining me on this explainer, and keep exploring.